घंटा होगा, 1 घंटे का रहता है बर्मा यहाँ का।
हाँ हाँ हाँ,
उस तरफ जाते जाएं।
हाँ जी, बर्मा पहले आ जाता है ना।
हाँ हाँ जी, हाँ। वन ऑफ माय फ्रेंड्स इज देयर, यूनाइटेड नेशंस में।
ओके।
प्रोक्यूरमेंट हेड विक्रम सिंह।
ओके,
इज फ्रॉम रीवा।
आई सी।
यहाँ कोई बैठा है क्या? कोई बैठा है क्या? अच्छा।
अब बताओ। हेलो।
हाँ विजय जी, मैंने YouTube पे कर दिया इसको। चालू हो गया ना? ठीक।
नमस्कार द्विवेदी जी।
नमस्ते सर। नमस्ते।
मॉर्निंग।
विवेक शिव जुड़े हैं क्या?
जी सर नमस्कार सर, कैसे हैं आप?
हाँ बढ़िया है। और शिव आप अच्छे हैं?
जी सर, आशीर्वाद है आपका। आप सुनाइए।
बस ठीक चल रहा है। बस आप लोग सब ऐसे जुड़ जाते हैं तो अच्छा लगता है
जी
ना?
सर। जी
आप
सर।
लोग
जी
ऐसे
सर।
उन्नति करते रहिए, बढ़िया आगे बढ़िए।
जी सर, धन्यवाद सर।
रुचिता, इसको स्टार्ट करें?
जी सर। So can I start? So
Okay,
start
good
करिए
morning.
आप।
Good morning to everyone. I invite to
आप
Dr.
करिए
Ruchita
स्टार्ट
Tripathi,
करिए।
organizing secretary of this workshop, to conduct this program. I request to all participants, please mute oneself. Ruchita,
जी सर।
please start your program.
So very good morning to all of you. I'm Dr. Ruchita. I extend a very sincere warm welcome to all of you present on the second day of this web workshop of our college. Proceeding onward to the following phase of this workshop, today's schedule is first of all, Dr. S.P. Shukla sir. He is coordinator of IQAC and mentor of NEC of our college will give the welcome remark. After this welcome remark, our guest speaker, Dr. Shiv Bharadwaj, he is International Research Professor, Department of Biotechnology, Yangnim University, Korea, will give the lecture on Introduction to Molecular Dynamics and Quantum Mechanism in Drug Discovery. I would like to invite Dr. S.P. Shukla sir for welcome remark.
Very good morning to all the participants and panelists and faculty members who are attending this international web workshop. It's a pride moment to have with us Dr. Shiv Bharadwaj, an eminent scholar, laureate, and international professor. On the second day of international web workshop being held at Government Thakur Ramat Singh College, an institute of excellence in higher education. As you all are aware that this international event is organized by the Department of Geology, Biotechnology, Botany and Microbiology in collaboration with National Academy of Science, Bhopal chapter on computer-aided drug discovery with natural resources. The previous day, we had two interactive and innovative sessions by Dr. I.P. Tripathi, Professor Pro VC, Dean, Faculty of Science, Biotech and Environment, who shared his expertise on computer-aided drug designing techniques with natural resources. The second session was held by Dr. Vivek Dharduvedi, scientist at Pathfinder Research and Training Foundation, Greater Noida, Uttar Pradesh, who delivered a nice lecture on the concept and application of molecular docking. As a director of IQAC, I am privileged to offer my heartfelt greetings and welcome to Dr. Shiv Bharadwaj today. On behalf of the college, my principal, Dr. Arpita Awasthi, the organizing team, Dr. Amit Tiwari, chairperson, Dr. Mahesh Shukla, coordinator, and Dr. Ruchita Tripathi, organizing secretary of this international web workshop. Dr. Bharadwaj is currently rendering his responsibility as a research professor at the Department of Biotechnology, Ingram University, Korea. He will deliver his lecture on Introduction to Molecular Dynamics and Quantum Mechanics in drug discovery. I would like to invite Dr. Ruchita Tripathi to say a few words about Dr. Bharadwaj and to start the session. Dr. Ruchita Tripathi.
Thank you, sir. So prior to beginning of the lecture, I might want give a short prologue to Dr. Shiv Bharadwaj, sir. He is international research professor from Yangnam University, Korea. Basically, his research work is focused on the discovery of bioactive molecule and green nanoparticle as potential inhibitor against microbial infection and disease in human, and how these bioactive compound alone or conjugated or nanoparticles affect the microbial pathogens and discovered tissue cells using assay development and bioinformatic data analysis also. His expertise in biological assay development and evolution using biophysical techniques and omics data analysis. Presently, his research included green nanoparticle synthesis for functional cosmetic and antimicrobial agent food pathogens. He is also work on computational added drug discovery followed by in vitro validation for infectious agent disease and bioinformatics sequence data analysis. As international research professor, he has initiated and established the nanotechnology and bioinformatics project research work in the Department of Biotechnology, as well as produce international collaboration on drug discovery and 3D cell structure. Additionally, he has produced several peer-reviewed publications in the field of nanotechnology and drug discovery. In the continuation of this session, as you all know, at the yesterday on the first day of our international workshop focused on molecular docking by Dr. Vivek Dharduvedi, he demonstrated practical based lecture on this docking technique where he has described which type of step used in this technique and what is the importance of molecular docking in drug discovery. Now today we proceed further and will be learn about molecular dynamics and quantum mechanics by Dr. Shiv Bharadwaj sir. So I would like to invite Dr. Bharadwaj sir for give the lecture on this topic.
Hello. Can I start?
Ji sir.
Okay. Very thank you, Dr. Ruchika ma'am, for wonderful introduction. First of all, thank you to the organizing committee, and thank you for giving me opportunity to present a lecture here and share my knowledge. Very good morning, all of to you. So let's begin today's section with the introduction of molecular dynamics and molecular mechanics in drug discovery pipelines. Can I share my screen now?
Yes sir.
Well, just 1 minute, please. Is it visible to everyone?
Yes sir.
Okay. So let's begin. Today's lecture is about an introduction to molecular mechanics and quantum mechanics in drug discovery pipeline. So basically it's going to be about on computational chemistry. If we talk about molecular mechanics and quantum mechanics, we study these two topics under computational chemistry. So basically what I'm going to do today, I'm going to cover these following points, where I'm going to provide you an overview on drug discovery process. Then we will define what is computational chemistry and how we can calculate different methods are like molecular dynamics and molecular mechanics under computational chemistry. Then we will look into what we can learn from computational calculations. Then we will have an overview how we can use this method in the computational chemistry and where to use them. Then we will learn what is the difference between molecular mechanics and quantum mechanics. We call them MM or QM in short form. Then we will learn how we use MM and QMM in drug discovery process. So let's have a view on drug discovery process. If we look in the history, what used to happen, this is one example I have considered here. In 1920, North Dakota, cattle were died because they have eaten some rotten clover. The disease is known as sweet clover disease. But when the researchers look into it, this matter, then they figure out this clover, the rotten clover, contain curamine, but it's converted into 4-hydroxycuramine by the fungi. And when this is converted by the fungi, it becomes a dicuraminol, which is strong anticoagulant. That was the reason behind for the death of the cattle. So what researchers have done then, they look into the incident, they check the hay, they check the pharmacological effect of the compound, they link those pharmacological effect of the compound, then they figure out that it has a strong anticoagulant activity. Then they check the chemical variation of the compound. Now we have that compound, warfarin, which is they use largely in different disease. For example, warfarin is used in thinning of the blood, or you can say that it is used in heart stroke. So that was an overview, historical overview, how the researchers took the advantage of accidental discovery to a drug. But now the whole process is changed. What is the process now? We have the disease. We look into the relation between the disease and the molecular mechanism. We see what are the proteins in a very general language, I will say. What are the proteins are involved in the disease? We know very well that proteins are coming from the gene, so we can look into the genes also. But here I will consider only the proteins. So we check the relation between the protein, how the protein or protein signaling, like yesterday Dr. Vivek mentioned about this protein signaling or cell signaling. So we look into this particular mechanism, how these proteins are interacting with each other, and then we look for the definition of a drug target. Definition of a drug target means we are looking for the particular protein which is causing the disease, right? And then then we have the protein target, or we can call it a receptor or a protein. Then we check the molecules which can inhibit it, this particular target. So yesterday, Dr. Vivek already had mentioned about it, where we can find the protein, crystal structures, and how we can screen them. He mentioned about the in silico techniques, but I'm not going to give a much detail on it. So it's going to be an overview. So then we have the compounds. After we have the compounds, we check the chemical variations of the compound. Chemical variation means we check how we can modify it. There are different kind of functional groups in it. For example, carboxyl group, OH group, hydroxyl group, or maybe ketone or aldehyde group. So we change these functional groups and then check the activity of the molecule, how the activity of the molecule or the structure of the molecule or the chemical properties of the molecule is going to inhibit the protein or our drug target. Taking that molecules, we go for the preclinical or clinical evaluations, and then we go for the approval, and then following we have this treatment of the disease, what we have targeted in our study. So if we look at on the drug discovery process, this is not a very short one, but it's a long journey. How? You can look here that, for example, we have 10,000 drug molecules. We have selected 10,000 drug molecules. So now these drug molecules can be natural compounds or from in-house research, or we can say that we have synthesized that synthetic molecules. We consider the molecules and then we do in vitro inhibition assay. That is a preclinical assay. In vitro inhibition, we see does the enzyme is getting inhibited by the compound or not. If it is inhibited, then we select these molecules. For example, if we have selected 10,000 molecules, but in preclinical studies, we have sorted out only 250 molecules which are showing some inhibition. The rest of molecules are showing no inhibition or no activity to the enzyme or the target we have selected. So following what we are doing, we are taking these 250 molecules or drug molecules for further clinical trials. These trials, if you are taking 250, most probably at the utmost, we will get only 5 molecules which are going through the clinical trials have positive results without any side effects. And then these clinical trials results together with the molecule go to the FDA approved, that is the panel. If everyone knows about the FDA, I don't need to give a definition of it. So the FDA will review these molecules and then select these molecules are compatible for the total human race or not. And then they approve it. So for example, if the 5 molecules are submitted for FDA approved, maybe different their norms according to FDA norms, maybe only 1 molecule get approved. So if you see in total that around 6.5 years are taken for only drug discovery and preclinical, then we have almost 7 years for the clinical one, and then 1.5 for the FDA approved. So you can imagine that, drug discovery process is not a short journey, but it's a very long journey. So let's take an example of corona. And now everyone's talking about why there is no drug against the corona. This is a reason because making a drug, new drug, is a very time consuming, and the picture is before you. So what is the convenient option for us against the corona? That we can choose the FDA approved drugs, and then we can do the in silico evaluation, and then we can save the time. For example, if we are taking approved drugs from the FDA, you can see that here only 1.5 years for the FDA approved. So maybe that whole process condensed to only 6 months or maybe 1 year, right? Because we don't have to go through all these clinical trials because the drugs are already approved under clinical trials. Then we have to see only the activity of the drugs against the coronavirus targets. So there are different targets in coronavirus. We can choose them. That's a different story. So now we have a long journey for the drug discovery process. So, but that's another thing only associated with it. We have different risks also associated with it. What are those risks? For example, we have target discovery. We have to choose a drug. We have to choose a target. Unless we do not have a particular target, we cannot blindly go and put the drug into the animals and then check that does the disease is going down or up. What is happening to it? So we have to find the target. That's the priority of the drug discovery process. Then we have lead discovery and lead optimization, where we are modify the molecule according to the functional groups and try to increase the activity and tries to reduce the toxicity. Following, we have preclinical, that is in vitro evaluations and other in vitro experiments. We are not using animals or humans. So in phase 1, phase 2, and phase 3 of the clinical trials, where in the phase 1 we use animals, and then rest of the phase we use humans, and then following, if we have the positive results, we are going for the registration of the compound and then FDA approve and all this. So if we see here that from target discovery to preclinical, we have only 24.5% of chances of success. And if we look into preclinical to the phase 3, that is the part of the development. So we have only approx 7% chances that the drug will be approved or drug will be produced, or we have some positive results. So if we have a total picture into it, so overall chances to produce a drug after spending around 15 to 20 years, we have only 1.8% chances. So this is the traditional drug discovery process, and then this is the drawbacks we can say associated with it. What is that? That if we are following this whole process, then we are getting only 1.8% chances that we can have the drug approved, or we have the particular target get inhibited, or we have a particular disease getting cured. Let's take an example of HIV. Why we do not have any drug? Because the HIV is getting mutated itself, and there are other certain factors which are coming from drug discovery process. What are they? These are the factors. So how we can overcome on these factors? So then we have this computer aided drug design process, or we can say computational drug designing, because computational drug designing reducing this 15 to 20 years to, let's say, like maybe 1 to 2 years, the process, the discovery process, where we are spending 6 to 5 years. So that's why we are now using this computational drug discovery tools, or the methods in the drug discovery protocol, or the pipeline. So now let's take a look into the expensive, how much money we are spending to develop only one drug. So I have put a chart here. You can see here from 1963, only round we have 15 molecules approved, and then money was spent in 15 billions. But if the latest I have here data is from 2013, if you see here that almost only 50 drugs are approved, and more than 45 billions money has been spent on it. So there's another factor that inspired us to use the computational drug discovery process to design the drug in the drug discovery process. So for example, now how the things are changing with the drug discovery process, also affecting our this drug discovery process. For example, it's now very difficult to find the targets because the disease are now these days very complicated. Let me put an example like this. If we are talking about the tumor, the cancer, now the biologists have already discovered that within the tumor have their own microenvironment, and even in the microenvironment, each cell has its own genotype. So if you are targeting one particular target or one particular protein, considering that the protein is there and then we are targeting it and then tumor will be cured, no, because the tumor cells have their own genotype and the protein getting mutated. Or if, for example, if we are targeting one target or one protein in the tumor, maybe the protein 2 of the second cell will express more and will enhance the cancer. So that's the reason that now these days we had different approaches to tackle the cancer, and then different approaches for development of drug. So second thing is that we have now chemical matter. Chemical matter means that now we do not have many sources for the drug discovery. I mean to say, like, for example, what we are doing in the drug discovery, we are taking the molecule and then we are checking it. But then we are doing some optimization, lead optimization. We are changing the molecule structure by structure activity relationships and something like that. But now the limited backbone, I would say, the backbone of the compound or the drug is limited. So now they are trying to manufacture new molecules. They are doing synthesis. How they are doing synthesis, that's the big problem. But the computational drug discoveries again assist is there here. How? Using artificial intelligence. You may heard about the corona in the corona drug discovery, they are using artificial intelligence where they are taking different properties of the molecules, and then they are checking the target's active site, and then they are checking how these different molecules and then different fragments of the molecules, we join them together and then we can make a single drug. That's a new approach in the drug discovery. We call it fragment-based drug discovery. We are not taking a big molecule now. We are taking different small fragments of the molecules, and then we are docking it on the active side of the protein or our target, and then we are calculating which fragment is showing more affinity, and then we are joining them together and then making a new molecule. So again, if we have to do this by chemical methods, by in traditional methods using in vitro, we cannot do it. So here again, we are using uh, computational drug discovery process, or I would say we are using artificial intelligence in the computational drug discovery process. And then we have biologicals. In the biologicals, we consider it sometimes antibodies and all. So we are checking sometimes, does this, not sometimes, but we need to consider it, does this drug do some side effects or not? So we cannot sacrifice many animals, we cannot sacrifice thousands of humans just for trials, human trials. So what is there now? Now the scientists are coming with the new ideas that we call some kind of synthetic cells, or we have some computational programs. For example, we have PDPK, or we are checking some kind of modeling using the computational tools, or in fact, we are using some computational tools. We are making the whole cell, and then we are putting the drug inside, and then we are using the simulations. We are checking how this protein, the particular target, our target is getting affected within the cell, within the whole cell, with the whole cell environment when we are introducing our drug molecule. So in total, we do not have much options left with the traditional biology or with the traditional drug discovery process. We have to now, this is the high time to, not high time, but this is the ultimate option left with us to include the computational drug discovery methods or approaches in the drug discovery process. So now what happens then? Now the drug discovery process is not a single field, it's a consortium of different fields. For example, if we are talking about the drug discovery project, we have to consider the medicinal chemistry, we have to consider combinatorial chemistry, we have to consider in vitro, in vivo, toxicology, upscaling, patents, pharmacokinetics, traditional medicine, automated assays, structural biology, and of course, then comes the computational chemistry. So among all these, computational chemistry also contribute. How it contributes, we are going to look into their two methods, particular methods, the quantum mechanics and molecular mechanics in this lecture. So before I proceed to the quantum mechanics and molecular mechanics, I would like to give you an overview on the chemistry. So just a brush up. We have chemistry. In the chemistry, we have periodic tables. We have F block, D block, P block, and S block elements. We have electronic configuration of the element, each elements, and then these elements comes together. So make the molecules, compounds, and then these compounds react to each other, and then we have chemical reactions, and we study them in the chemistry. So let's take an example on the molecule. So you can see here the 3D molecule. 3D molecule I have put here for your consideration. You can see here, if we make a 3D structure of the chemical compound, it looks like this. Then we have molecular orbitals. Molecular orbitals are, if you are familiar with it, the molecular orbitals are the space around the nucleus in an atom where is the probability of finding the electron. Then I have put an example here, the molecular electrostatic potential. It is, I will explain detail in later, but just an overview here that this is providing the electrostatic surface of the molecule. If the molecule have a charge, how it would looks here, I have showed here. So now what is the purpose of showing here 3D structure and molecular orbitals and electrostatic potential here? Let me tell you, the 3D structure is going to be, we are going to consider it under molecular mechanics. And these molecular orbitals and molecular electrostatic potential, we are going to consider them under quantum mechanics. So we need to consider here before we proceed further that in the 3D structures, when we are talking about the molecules, not the atoms, when we are talking only about the molecules, we use molecular mechanics. But when we are talking about the electrons, why? Because we are talking about the orbitals, and orbitals are what? Where there is a probability of finding the electrons. So when we are talking about the electrons, we always say about quantum mechanics or quantum physics. These are two interchangeable terms, correct? Okay, so let's begin with the computational chemistry. What is the computational chemistry? It's a branch of chemistry where we are using the equations to figure out how the matter behaves at atomic scale and how we can use the computer to solve them. Also, we calculate the structure properties of the molecules, and these molecules may be in solid, liquid, or solid state. And then we also use this computational chemistry to explain or to predict chemical phenomena. For example, reactivity. We can use this computational chemistry to figure out the reactivity of the molecule. Then we have very high accurate calculations, for example, ab initio method, to less accurate semi-empirical to very approximate molecular mechanics. What does it mean? It means that ab initio method to semi-empirical methods, we are considering the electrons. If we look into the matter, matter is made up of molecules, and molecule itself made up of atoms. And these atoms come together and they share the electrons by either bonding covalent sigma or pi bonding. They are sharing the electrons and they are making the whole structure of the molecule. So if we are talking about only the molecule, then we are missing the electrons. We are missing the nucleus. We are missing the I would say, uh, sorry, someone saying something? Okay. So if we are talking about the molecules, then we are missing the electrons, and then we are missing the small further particles. I'm not going deep into the physics here. So ab initio methods are more accurate. Why? Because we are considering electrons, and electrons comes together and make the molecule. So if we know about the electrons, then we can easily give information about the molecule behavior. Correct? But if we know about the molecule, but we cannot give the information about the electrons because we don't know about the electrons there. So that's the difference. So when we are trying to figure out the chemical reactions, we always consider the electrons. We are not considering the atom. We consider that how many electrons are transferred from A to B. So that is how we define the reaction. So that's the same thing here. We use ab initio method is the whole thing. They are talking about the electron, motion, spin, orbital, and everything. So that's the ab initio method. In the molecular mechanics, we are talking about how these atoms are moving in the space. We will look into this picture in the next slides. So what is computational chemistry then? It's a mathematical description of the chemistry. So how we can, for example, we are saying that A and B coming together, reactants making the product under the catalyst C or D, whatever. So this we are writing down on the paper. So that's what, this is a mathematical description. So computational chemistry is a theoretical chemistry part. And then second part is computational chemistry. What we are doing in the computational chemistry, we are taking this mathematical description, what we have written in the paper, we are putting it in the computer, and then we are computing it, we are simulating it, and then we are checking does the hypothesis we have generated in the mathematical description or theoretical, we have theory proposed, or we have proposed the hypothesis, does it works there given the conditions and what we have made. Let me tell you, before we have the computational chemistry, scientists used to do all these calculations by hand on the paper, and then they are piling thousands and thousands of papers, but it was very time consuming. A single small mistake used to have spoiled the whole hypothesis. But now with the technology we have today, that's a computer. Now even we are going to have advanced computers, that are quantum computers. The drug discovery process is going to be very less. For example, now in previous slide, I have mentioned 15 years, but in maybe in coming years, we can produce the drug in within 1 hour. That's a different story. But let me tell you here that computational chemistry, we are considering the theoretical methods, and then we are considering the computational coming together. How? We are taking a chemical problem, we are making a physical methods for it, and then we are generating a mathematical description, then we are doing simulation, and then we are checking it does the problem we have generated is giving us the same results or not. Does our hypothesis fulfill here or not? So what computational chemistry includes? If we look into computational chemistry, there are some similar methods that seems like to be in the computational chemistry, but they are not. For example, time-dependent ab initio calculations. When I say ab initio, that means we are talking about some computer calculations using theoretical models. So how these theoretical models behaves under the time, that comes under computational chemistry. Electron dynamics, how the electrons behave with respect to the time or in the space, we study them under computational chemistry. Then of course we have classical molecular dynamics, that sometimes we call it Newton physics or Newton's dynamics also. We have semi-empirical calculations. In the semi-empirical calculations, we consider some half experimental data and half theoretical one. Then we have some selected models. Then we have cross-grain models also. These all models included in the computational chemistry. But what is not included in the computational chemistry is quantum chromodynamics, quantum models, okay, calculations on gallium, computational fluid dynamics, data mining, and rule-based derivations. This is just an overview to tell you what is in the computational chemistry and what is not in the computational chemistry. So what is the advantage of computational chemistry? So computational chemistry allows us to use the computer power to measure the following properties for a molecule or any biological identity. If we are talking about the biology, if we are talking about the chemistry, then we are talking about the chemical species. So what we can do, we can calculate the molecular geometry. We can calculate the electron density. Electron density means we can calculate the orbitals. In the orbitals, we have frontal molecular orbitals, or we can calculate natural bonding orbitals. Then we have electrostatic potential. Of course, now electrostatic is a charge, and the potential is how its distribution on the molecule. Then we have conformational analysis. We can check the arrangement in the space, and then we can calculate the different types of energy, the Coulomb, London, or van der Waals. These energies can be calculated using computational chemistry. Another thing is that we can determine the structure of the ligand and the protein through X-ray crystallography and NMR spectroscopy. So here, these are the physical methods, but if we have computational techniques, we can also generate these structures and then we can study them. Then we have receptor and ligand docking that yesterday you have attended, and then we can exactly measure how the geometry and the energy favors such ability of such interaction. How the energy is participating in the stability of these compounds, means receptor and ligand. Then we can compare the various ligand through various parameters. For example, we can, in the drug discovery, mostly we consider binding energy. In the binding energy, we have two different methods like MMGBSA and MMPBSA. These are two different methods, and beside that, we have other factors or energy factors to consider that we can consider. So what does computational chemistry calculate? It calculates the energy, structure, properties of the chemical system. For example, for a given geometry, what would be the energy? For example, I will later explain that potential energy for the molecules we consider in quantum mechanics, and how this energy varies when the geometry changes, and which geometries are stable. We consider the geometry is stable when the imaginary frequency is 0 there. Then we consider how do atoms rearrange to form new molecules. That is how the atoms comes together and make the bond and do the chemical reaction or make new molecules that we study again in I should mention here that is quantum mechanics, molecular mechanics approach. There's a hybrid approach, but we are not going to discuss that particular approach in this particular lecture. And then we have how do the structure, energy and property change over time. That is time dependent conformational energy changes of a particular system, I would say here, or you can say that molecule or chemical species. So what else we can do with it? We can in fact calculate the enthalpy. We can calculate the dipole moment. We can orbital energy levels like HOMO, LUMO. Then we can calculate the ionization potential or energy. That's the HOMO energy. Electron affinity. That's the LUMO energy. We have electron distribution or electron density. Then we can calculate proton affinity, electrostatic potential. Even we can calculate the vibrational frequency and normal modes of any molecule. We can calculate electronic excitation energy also. We can calculate NMR, chemical shapes and coupling constants. Even we can calculate reaction path and barrier heights and the reaction rate. If we look from this vibration frequency down there, we can perform all these reactions in the computer rather than going and using expensive instruments. If we are using a proper, a very high accurate method, or I would say model here, our basis set, our theoretical model, we can exactly calculate or very approximate, we can calculate these properties using computational chemistry. Then we don't need to go and do physical one. Yes, of course, if you are calculating here, then you can go and do the physical methods or these physical techniques you can use to validate your method. So what we are going to learn from the computational calculations, we can learn the molecular visualization of the molecule you have seen yesterday. Then we have molecular mechanics, that's classical Newtonian physics again. We have semi-empirical molecular orbital theory, ab initio molecular orbital theory, density functional theory, geometry optimization, and molecular dynamics. Now, if we see the semi-empirical to density functional theory, these three particular part considered under quantum mechanical methods, and the rest under molecular mechanics. So, how to conduct a computational project? If we want to initiate a project on computational chemistry, how we can initiate it? So the question should be answered before we select a project. What do you want to know about a chemical species? For example, I want to calculate the chemical reactivity. So I have to opt quantum mechanics, not the molecular mechanics. How then the next question comes, how accurate does the prediction need to be? It depends again now. For example, I want to spend more time. I have a very good computer configuration with me. So according to that, I can select the method. And then it comes prediction. So prediction means that how approximate. Let me tell you one thing here, that in the computational chemistry, whatever we get, it comes in approximate. So let me define you approximate, and let me correlate with the biology here. So in the biology, whenever we do any experiment, we always do it in replicates or try to do as many duplicates we can do. Why? Because we want to get rid of the standard error there. If we are taking only one experiment and then taking the reading from there and then try to establish our theory or our hypothesis with the outcome, it's not possible. It's not validated. This is statistical analysis. We cannot do it. So similarly here, how accurate the prediction. So that means how many times you want to titrate your hypothesis in the system. It means that if you want very accurate one, then you have to do like in biology mostly we do triplicates. If we want less accurate, then we are doing only duplicates. And if you want more accurate, then we are going at least 6 replicates. That's a very accurate one, because here if you are taking at least 6 replicates, then the standard error or the standard deviation will be low. Similarly here, if we are doing the titration in the computer only 2 times, then we have less accurate. But if we are doing titration in 20 times, then we have very approximate results. So approximate here means that we are trying to get rid of any error that may originate during the calculation in the computer. So that is how accurate does the prediction need to be, or you like to have. So then next thing, like I mentioned in the first point we heard here, how much time you can devote it to the problem, how much time you have. For example, your project is only 1 month time, but you have selected the problem which is not going to be complete within 1 month, but rather in 1 year. So that's not going to work for your project. Then what approximation are being made, I just explained before that we are doing some titrations, and then it depends upon the method how we are going to do this titrations or replication, or how many times we are going to do it. So the answer to these questions will determine the type of calculation, model, level of calculations, or basis set to be used. So do not get confused with the level of calculation and basis set, because I am going to explain them one by one in the following slides. So here we comes with the types of computational chemistry calculations. There are many types of calculations, but I have selected some few one which is related to the drug discovery one. So to make you a bit easy. For example, the 3 basic types of calculations that can be done in the computational chemistry to figure out some information that are single point energy, that is predict stability and reaction mechanism. Second is geometry optimization to predict the shape. Then we have the frequency. Of course, everyone knows about vibration and frequency to predict the spectra. These 3 things we calculate to calculate the property of a drug molecule, and sometimes protein also, but not protein if we are using it not a target, but if we are using protein as a antibody. So during that one, we can use these computational methods on the huge protein. But let me tell you that if you are taking a huge protein, or huge number of atom collections is going to be very expensive. Why? In the following slide, will explain to you. So mostly we consider these computational methods depend upon the method. We may choose the type of the method we would like to apply. For example, for the protein, we normally use molecular dynamics, but if we are talking about the ligand or some small atom collections, we may use quantum mechanics. So depend upon the type of the method and the starting point theory, we may either consider classical method and or the quantum chemistry method or quantum mechanics. If we look into a classical method, these are the methods use quantum mechanics to model molecular system. For example, we have molecular mechanics and we have molecular dynamics. So some of you may be familiar with this molecular mechanics and molecular dynamics, but I will give you a little brief introduction about it in the following slides, and even the quantum mechanics different methods. But let me tell you again, repeating here, that if we are talking about the molecule, then we are talking about the Newton physics or Newton mechanics. So that's the classical method. But if we are talking about the electrons, then we are considering quantum chemistry methods in our calculation or in our selected problem. So now we have a question here, what is computational model? So when what you call computational model, a model is a system of equation or computations used to determine the energetic model of a molecule. That means what we are doing, we are scripting the problem, we are making equations. For example, a plus b square is equal to a square plus b square is equal to 2ab. That's the equation. That's the model now here. For the computer, that's the model. That's the equation. Now we are using different models, use different approximations or level of theory to produce the results of varying level of accuracy. For example, now if we want to measure the distance between two the atoms, we always measure it in angstrom. We are not going to measure it in kilometer. That's a stupidity. So similarly, we have to select the approximate method or the level of theory which will give us a accuracy in our model. So that's the computational model again that we use it. So there is a trade between accuracy and computational time. So what happens in the computational model? If we are choosing a very accurate model for the calculation, then the computational time increase and the computational cost increase. I will explain in the following slides how. So there are mainly two types of model that we use. Those use Schrodinger equations and those do not. Now you may be wondering about what is Schrodinger equations here. So let's check it in the following slides. But these are the characteristics are the parameters that we define a computational model in the computational chemistry. So types of computational models. If we put these models from least to most accurate, because we want to calculate something for ourselves. So if we put from the least accurate, we don't want very accurate results. We want very approximate results, okay? Or maybe less accurate methods, results. So according to that, we can classify our list time that we have molecular mechanics. This is at all use of Schrodinger equations. In the molecular mechanics, what we do, we calculate the empirical energy function parameters against the experimental data first, are simply generalized, not very accurate. That is, we are normally using only atoms 1 to 104 atoms. Less than 104 atoms we are considering in molecular mechanics. In the semi-empirical molecular orbital method, this method, we use the experimental parameters. That means we have done some X-ray or NMR studies, and then we take this one, and then we mix it with the Schrodinger equation, and then we produce some data. So semi-empirical method or semi-empirical molecular orbitals method, what we are doing, we are taking half of little part of our experimental data, and then we are mixing it with some computational equations or computational models, and then making a new hybrid one. So why? Because it's a semi-empirical, because we have some experimental data here. Then we comes with the ab initio molecular orbital methods. It uses the Schrodinger equations, but with approximation. What does it mean with approximation? That means that we are not going to very accurate one. We are just touching the average, on an average, we say. So these are very computational expensive one, and accuracy is bit more compared to the semi-empirical one, but we cannot consider more than 10 atom in it. Then we have density functional theory, which is again using the Schrodinger equation with a consortium of other theories like in ab initio methods. I would say like ab initio method and density functional theory is on the same level. Because they are using the same equation. Schrodinger equation is the core one, and then there are certain other theories suggested by the physicians, and then some theoretical physicians, and then chemists and all, and then they make approximations or make some methods to study the atom behavior within a molecule. So according to that, they have differentiated here. So in the density functional theory, what's the difference between differential functional theory and ab initio method? That ab initio method is very expensive, but compared to ab initio method, density functional theory is less expensive and then more efficient method. That is, you can see that we are using almost same number of atoms, but accuracy varies with respect to the level of accuracy we want to like have. So in the density functional theory, like ab initio method also, we can select the different layers of accuracy what we would like to have, and depend upon the accuracy, depend upon the level of this model within the density functional theory, we can approach more accuracy. So you was looking at here the Schrodinger equations, but if you don't remember about the Schrodinger equation, so let me give a little overview. Schrodinger equation, Schrodinger equations suggested by the scientist Schrodinger. It says that the light has a dual behavior. That means it can act as a light. It act like a wave and then particle also. So this how they do it, there's a lot of theory behind it, huge literature available. You can easily find it on the internet. I will not go deep into it, but the Schrodinger equation, he considered that there in the light there are certain small particles, he called them photons. Now we know that these are electrons, or even we can break down electrons or nucleus into sub atomic particles. So according to that, according to him, these small particles when fall on the surface, they get diffracted, and then there was some diffraction phenomena, which led to the theory of this, that light has dual behavior, that it act like a wave also, and it like a particle also. So now we have types of computational model. I'm going to focus on molecular mechanics and this quantum mechanics and molecular dynamics. So if we look into the molecular mechanics, what is molecular mechanics? A model based on classical mechanics in the simplest type of calculation. What it do? Here we used when the systems are very large in a macro size, not an atomic scale. And approaches, we use approaches that are more accurate, become too costly in time and memory. That is, we are not using very expensive computational cost systems or models here, but it's approximately the cheaper one. We use the information in the molecular mechanics like bonding, stretching, bonding, bending, torsion, bond torsion, I would say, electrostatic interaction, van der Waals forces, and hydrogen bonding to predict the energy system in a molecule. Let me tell you that I am not considering the here mathematical equations here, because if I'm going to mathematical equations, then you may be puzzled and then you do not understand anything. So I have tried to make it as simple as I can, because most of you must be from biology background. So I have only mentioned here about the terminologies, but I'm not using any mathematical equation. So if you want to have some mathematical equations, you can easily find on the internet. If you have a little bit idea or introduction about molecular mechanics or quantum mechanics, then it will be easy for you to understand the advanced level in it. So the third point is that the energy associated with a certain type of a bond is applied throughout the molecule. This leads to a great simplification of the molecule. So what we are doing here, we are applying the energy to a molecule. So like I said, the molecular mechanics is based on classical mechanics. The classical mechanics means the Newton law that says that force is equal to mass into acceleration. If we are applying the force on a particular mass, it will accelerate. The same approach, the same principle we are using in molecular mechanics. So it should be clarified that the energy obtained from the molecular mechanics do not have any physical meaning, but instead describe the difference between varying conformation type of isomer. For example, in the following figure, you can see the two walls are connecting with the one string. We are applying the energy on the both. What is happening? They are changing the conformation. They may vibrate in the space. Similarly, if you see the three body here, you may say that they make a different conformation. They may rotate in any direction. So if we consider another example, that is a spring is attached to the one side and then loaded with the weight. So it stretch and then come back. So that is stretching again. So what is happening then? These different macro molecular physical processes, I would say, we consider them under molecular mechanics. How these physical changes happening in the molecule, these are considered under only molecular mechanics. So molecular mechanics also provide us the heat of formation if the zero of energy is taken into account. If we are taking that our system do not have its internal energy, of course, then we can provide the heat of formation in it. So what does it mean? That molecular mechanics we can apply only to the very big systems, but the results are not always very reliable. But if we are using some other level of accuracy, then we may increase the accuracy in our computational model under molecular mechanics. How we can do that one, I will explain later in following slides. How? I said that we are applying a force. Force is equal to mass into acceleration. So we have force field. So force field what we are doing, force field is nothing but it's a collection of parameters about how the bond stretching happens, how the angle bending, how the torsion angle and the non-bonding interactions happen within a molecule. But let me tell you very clearly that these force field like bond stretching here should be is it's very specific to the uh, for example two atoms. For example, I'm talking about bond stretching. So bond stretching have a particular stretching level based on its physical identity or conformation in the space. That conformation or bond stretching cannot be same to the C with the carbon, carbon with the oxygen. So carbon carbon bonding, carbon oxygen bonding, they have two different bond stretching. Why? In the one we have only single bond, if we say, in the other one we have double bond with the oxygen and carbon. So these similarly like angle bonding, torsion angle and other certain factors, these are different for the different molecules. So we make different calculations how this bond stretching happens. There are different equations and then put them together and then we make one a parameter or one equation and then we put this equation in the system in the computer and then apply this approximation of our hypothetical parameters on the molecule and then we check how the molecule behave under these parameters are our given force fields. So thus you understand what is force field. Now force field have one drawback. The force field ignore the electrostatic distribution, but quantum mechanics consider electronic distribution of molecules. So now recent studies have also established ab initio molecular mechanics or dynamics, but we are not talking about here. And even we have ab initio or polarized force fields where they are considering electronic distribution also for the molecule, but the computationally very expensive. And then very occasional cases are in a very special cases, these polarized force fields are being used. Or else we have other examples. For example, for, in drug discovery, mostly we are using umber or we are using charm or we have OPLS also. So what is umber? Umber is assisted model building with energy refinement. And if we talk about the charm, it's a charm is a chemistry at Harvard molecular macromolecular mechanics. Under umber also, for example, let me tell you that umber, the very force field comes was UFF, that was universal force field, because they do not know about the molecule. They consider that this molecule have this approximate this level of stretching, this approximate level of bonding. So this UFF is one particular force field. Then they have different, like now we have they have FFO2 pole or FF12. These are polarized force field in the umber package. So if we see that what now, since the biological system, let's talk about the protein and only in inhibitor. So they may compose of like in protein they have residues. Residues mean amino acids, 20 amino acids, but in case of ligand we have these elements, carbon, nitrogen, sulfur, or sometimes fluorine also, and then oxygen. They come together and make this molecule or the inhibitor. So depend upon the molecule which are taking, we now have so advanced technology or so advanced force fields that for a particular molecular systems we have approximate force field or more accurate one. For example, if you see here in the table, umber, under the umber we have this FFO2 pole or FF12 pole. We can use these force fields on proteins and nucleic acids. The software which can provide you, or you can use this force field in the program, that is the umber. Then we have AMOEBA, where we can use protein, DNA, RNA, and lipids also. The program we can use is Tinker, Tinker HP, umber, or OpenML. Then we have a charm root we can use for protein, DNA, lipid, carbohydrates, and the programs are charm, NAM, GOMAX, and OpenMM. And similarly like this, we have ABBM, sigma pi, which is we use for the protein, nucleic acid, and Tinker. Then you may be wondering why we have different kind of force field for the protein when we have umber, or we have charm root for the protein, and we have umber for the protein also. The reason is that each force will have its own drawbacks. For example, if we, like I said, that the very first in the force field, that was UFF, if we consider that one, that was very generalized one. It was not specific about what is the stretching between the carbon-carbon, very accurate one, what we know today of using the physical techniques, that was known before. So that way, if we are using that particular force field for the protein, we will have some results, but not accurate one. So we will have some results, but not accurate one, but I will say that a very average one, or maybe below the average. So with the advancement of science and how the physicists and the chemists, they studying the molecule, and they are studying the molecule behavior in the laboratory, and based on this physical methods or their experimental data, they are designing these models. They are designing how much should be the stretching, bond angle, and all. Based on that one, they are making these formulations, for example, Lerner-Jones and other equations. And then based on that one, and these force fields are getting improved. For example, now if we talk about the charm, before charm has 26, now at if we see, I think it's 36. What happened between 26 or 28 and 36? They upgraded the force field. That means now, before, if charm is being applied only to certain molecules, certain molecules between carbon carbon or nitrogen is valid for that one, but now it is uh, more applicable to, let's say, DNA also. I'm just explaining, try to explain to you what's the difference between different force field, because it's very important when we go for the molecular dynamics. Later I will explain you what is molecular dynamics, and then we use this force field, or even in the molecular mechanics where we use the force fields, and then if we are using the wrong force field, then we will have some results, but not up to our expectations, or maybe not very accurate one. So then another thing is that in the force field, that these force field have a drawback of only number of atoms. You have been seeing the previous slide that we cannot use more than 10 atoms or more than 100 atoms. But as the force field getting updated, we may use them to the larger collection of atoms. For example, if I put an example on this Schrodinger program, there is a Desmond program where we use the molecular dynamics using Desmond program. But this Desmond is mostly used for a small molecule. If see there, they have given the name small molecule drug discovery. So if we are using a protein of 100 residues, then the OPLS 2005, before that there is OPLS 2003 or 2000. But if I say about 2005, this force field is very accurate to the small protein molecules within 101. If you are using a protein, and if you are using a half 100 residue protein, and then if you're using OPLS 2005, the protein will behave more accurately, and then the behavior will be, the results would be more accurate compared to other force field. But if you're using more than 100, then maybe your accuracy or approximation will be a bit down, but it's not like that you cannot use, but you can use. But if you want more accurate one now these days, based on the experimental data and all, then everyone says that the charm 36 is the more accurate one for the more higher set of atoms, that is beyond like say 500 or something like that. So based on the collection of atoms, we have to again consider our force field. Correct. Then we have application of molecular mechanics, where we can use this molecular mechanics. We can use this molecular mechanics to speed up the method, to open a wide range of systems. For example, we want to study the liquid behavior or liquid crystals or glasses, that is amorphous condensed phase. If we want to study their behavior, physical behavior, we can use molecular mechanics. Then in the biological systems, we have biopolymers like carbohydrates, membrane. Of course, you know about mosaic fluid model. We can use molecular mechanics. Even in the nanotechnology, for example, nanomaterials and molecular machines. In the nanomaterials, we now this uh, we have certain materials we used in electronics. So we want to check the behavior there, how they are behaving under electrical field and all. So we use molecular mechanics. In the molecular machines, we are using these molecular machines for different purpose. For example, how this protein pump in the membrane in the cell works, or how this ion channels work. So something like these small machines, we consider them molecular machines. Or some, we have artificial molecular machines also to deliver the drug also, or we sometimes we call them nano robots also. So such entities, we consider them under molecular mechanics. We have to study their behavior in a particular environment. So fast calculations allow large number of conformation to be screened. For example, drug docking analysis, global minimum search. So what are these two factors? When we are doing docking, as Dr. Vivek mentioned yesterday, you have to prepare the protein. You are adding the hydrogen and then you are adding a charge. But he also mentioned there that when we are doing the protein preparation or something like that, there's we use some force field. You maybe you have not observed it carefully, but when he was adding a charges and all, there is a force field. Then we see that the force field, what it do, it minimize the structure, correct? It minimizes the structure. That means maybe in the crystal structure there are some alternation, because the crystal structure may be some point error. So we are fixing it using molecular mechanics in drug docking analysis. Then we have global minimum search. For example, we have a ligand and then we don't know exactly this is the conformation for it or not. Why? If we have a crystal structure, then okay, there are some point error. But if we do not have a crystal structure, if we are making it in a chem draw or using other 3D drawing program or chemical drawing program, so for example Avogadro, it could be chemical or nanoparticle. So we are drawing in the computer, but we don't know exactly how it exist in the nature. So we again applying the molecular mechanics approach here using the force fields, and then we are trying to figure out the minimum or optimum conformation, correct? So if we have to do these analyses, we use again molecular mechanics. Then statistical data can be calculated. For example, if we have different molecules, we can do some statistical analysis also. For example, we are calculating root mean square deviation in molecular dynamics, but that is the advanced version of molecular mechanics. Dynamics with respect to time. I'm coming to that one also. So we also can do simulation of finite temperature, behavior, and phase changes. If we have to check them, we can also check them under molecular mechanics, and then we can apply our statistical data analysis. You know very well that biological data is nothing without statistical data. Unless you do not apply statistical analysis on it, we cannot validate it, correct? So these are the factors need to be considered. So the molecular mechanics, the molecular dynamics. So the molecular dynamics, what is happening that we are checking these molecular mechanics are different parameters with respect to time. So when we do with respect to time, that is automatically become our molecular dynamics. So what are the different molecule different softwares we can use? So we have VMD. And the VMD is a visual molecular dynamics. We can visualize our trajectory and all. What we receive from for the molecule behavior with respect to time in the molecular dynamics. Or we can do dynamics also, because it has a setup for the normed here. You can see here on the 6th, 7th options here. So norm, that is scale level molecular dynamics software. So we can plug in and then we can use it. Then we have Moldy, we have Gromax, Gromus, macro model also. We have Thinkers, Label, and Explore, and DNA tools. And we have even Desmond, which is a paid one. We can even use that one. So what is happening in molecular dynamics? Molecular dynamics is not much differ from the molecular mechanics. The difference is only that in the dynamics, we are continuously checking the behavior of the molecule with respect to time, how it is behaving under the force field. That's of course coming from molecular mechanics, how it is behaving under the force field in the space, and what is happening to its conformation, that we study under molecular dynamics. So I have already explained the Schrodinger equation here before, but if you want to have more look, so it's very important you should understand it before you jump to quantum mechanics. So Schrodinger equation, this Hamilton equation, Hamilton operator, it says that is equal to energy. That's a very simple form of Schrodinger equation. So Schrodinger equation is the basis of quantum mechanics. We already know about it, and it gives a complete description of the electronic structure of a molecule. We checked it in the previous slides. So if the equation could be fully solved, all information pertaining to molecule could be determined. If we can solve this equation for the whole molecule, we can check the electronic properties, or you checked before, we can calculate all those properties for a given set of molecules. But I should mention here that the Schrodinger equation is not 100% accurate to multi-body system. Multi-body system means that if we are considering more than one atom, we cannot accurately calculate all the parameters for each atom in it. That's a drawback in Schrodinger equation, and that's why we are always saying says in quantum mechanics that approximation that we have accuracy and all. That's the drawback. Why? Because the Schrodinger equation is applicable to only single body, that's the hydrogen atom. If you want to calculate the Schrodinger equation for the hydrogen atom, you can calculate each and every property for it 100% with accuracy. But if you are considering multi-body or double body or something like that, mostly we say that multi-body. So if we consider for multi-body, it's not very accurate, so we consider the approximation. So that's why we have a complex mathematical equations to completely describe the chemistry of a molecular system. Why? Because for the single atom, it's easy. We know about single atom, this, but if you're considering more than atoms, automatically become very complex one. Like I already mentioned it, that not solvable for a system with many atoms, that is multi-body system. Due to difficulty equation, computer are used in conjugation. Of course, if we sit down to write our own, we cannot do it. So that's why we are using the computers to understand or do this such huge calculations with the simplifications and parameters to solve the equation. So we use some simple parameters. For example, up here you can see that this is a Hamiltonian operator, which give us the potential energy with respect to the motion of the particle. But if you see here, it's a huge equation, but I have made it a simple, very simple one. So very simple one, if we put in the computer may knows that on the background, the H is what it means. It is a huge calculation on the back, but for us, it's only simple one. So that's a simplification we can introduce in the computer. So what next we can do using the Schrodinger equation, and that we already know very well, that it describes the wave and particle behavior of the electrons. Why we need this particle and the wave nature of the electron, I will explain later. For example, the wave function is described by the psi, while the particle behavior is represented by the E. Then in a system, more than one electron, wave function is dependent on the position of the atom. This makes it important to have an accurate geometry description of a system. So what happens that why particle and then the wave behavior? So if you remember in the previous slide, I mentioned about the potential energy of the molecule. Potential energy of the molecule is the optimum or global minimum conformation of a molecule or a set of or a collection of atoms in the space where they have zero frequency. So if a set of atoms have a zero frequency and then they have on a particular potential energy corresponding to no imaginary, I would say no imaginary or zero. Zero means frequency zero. If in the system, if the frequency is in negative, we call it imaginary frequency. So if we have a zero or no imaginary frequency and at a particular potential energy, So we call that the system is at equilibrium or optimum, correct? So we use this approach, this equation, to solve the wave function for the whole particle and making this whole molecule. So it's very accurate, and it's very important to calculate these two parameters for the selective system. So then we have semi-empirical molecular orbital methods. So what happened in semi-empirical methods, we use experimental data I mentioned before to parameterize some equations. For example, I mentioned that we have a set of data which explain what's the difference between how much distance between the carbon oxygen, carbon nitrogen, and something like that. We have a huge collection. Then we have, corresponding to these two atoms, we have torsion also, how the nitrogen moves in the space when it's bound with the carbon or the nitrogen, or oxygen, anything. We have many possibilities, permutation, combination we can do. We have these calculation data from the experimental data, and then we put them, and then we say, okay, if this is the distance, and if this is the torsion, how would be the inertia of the molecule in the molecule. So all these calculations we do, and then we put them with our theoretical data. So we do some calculations. For example, here I'm talking about semi-empirical molecular orbitals. So that means we are calculating the molecular orbital. Molecular orbitals means where there's a probability of finding the electron. So if we find the electrons, then we can exactly figure out the electronic properties, and electronic properties means we can exactly tell the true nature of the molecule. So here comes with the Hamiltonian and the wave functions. We used such equations to get the approximates, or to get some more accurate data, or to eliminate some errors. So, but semi-empirical method I have already mentioned is less accurate than ab initio method, but it's much faster. So if your requirement is that you don't want very accurate results, but somehow approximate data, and you do not have much time, then you may go for semi-empirical molecular orbital methods or semi-empirical methods. Then we have these equations are parameterized to reproduce specific results. That means there are less chances to get some error. Usually the geometry and the heat of formation. So that means if we want to calculate the geometry of the molecule, that means at a potential energy, potential surface energy of the molecule with the zero, or with the no imaginary frequency for a molecule, or a molecule with a specific conformation or optimum conformation of the molecule, we can calculate using semi-empirical method, because the data we repeat it and we get the same data. Similarly applied to the heat of formation. But these methods can be used to find other data. For example, now we have the geometry, we have the heat of formation, then we can calculate other properties. For example, I take the geometry. Now I have a specific geometry for my molecule. Now I can, let's talk in terms of drug discovery and terms of docking. So if I have this molecule with a specific conformer, so if you do the docking, during the docking, the conformation will not change. Why? Because it's already have attained its optimum geometry, correct? Because if it goes up and down, that's the negative or plus sign to its geometry. So at particular geometry, how is going to bind with the active site of the protein? So there you will get more accuracy about the inhibitory nature of the molecule or your inhibitor. Why? Because you know that it's not going to change, and how it's going to interact, or what kind of interaction is going to make with the residue at the active pocket of the protein. Correct? How? Because you know the geometry, this is not going to change. So similarly, you can do other calculations also from this, if you have some results from this equation or this particular computational model. Then we have ab initio molecular orbital methods. So ab initio molecular orbital methods, what happens here? As it is translated from the Latin, that the very first principle. So sometime we call it the first principle also. This refers to that no experimental data is used. That is completely theoretical one, and computations are based on quantum mechanics. Here now we are not considering the molecular mechanics, but we are considering the quantum mechanics. That means under quantum mechanics, we study ab initio molecular orbital methods. So what happens in ab initio molecular orbital methods? Here we are using mathematical approximations. Now you understand what is approximation. If I'm saying we are not getting the 100% accuracy. Why? Because 100% accuracy in quantum mechanics is applicable to only single body, but when we take more than when we take multi-body, then we cannot get the 100% accurate result under quantum mechanics. Correct? So that's why we are talking about here approximation. So mathematical approximations are usually a simple functional form, a very simple functional form for an approximate solution to a differential equation. For example, wave function. I showed you before that we can make a simple form like Hamilton is equal to energy E. We have operator. So, but if we look into deep, is a very huge one. So that means we can make a simple equations in it. So, but in ab initio methods, we have different level of calculations. What are they? We have Hartree-Fock calculation. We have molar passage perturbation theory. Sometimes we call it FEP, FEP calculations. We have density functional theory. We have configuration interaction, that is CI. These, if we talk about these, the more MP and DFT-CI, they consider the electron correlation, how the electron distribution is happening, and how are we say that how is the distribution on the distribution of the electron on the in the actually orbital, and then these calculations consider this electron cloud as a single body, because if they consider it as a multi-body, then it becomes very complicated to reach the approximation. So these equations or these models under quantum mechanics are popular because they are considering the electron density or the orbital as a single body during the calculation, and because of that they can easily define the behavior of the molecule, and is computationally less expensive and very simple one, like I mentioned here, the usually simple functional form. So let me tell you about Hartree-Fock's HF. This is a very simple less ab initio equation, very simplest one. But here the Hartree-Fock theory, what is happening, it's ignoring the electron distribution or the orbitals. He is just considering the nucleus. That's why he do not have very accurate results, which later get more additions. And then we have right now what we call DFT or CI interactions, higher level of calculations under quantum mechanics. Correct? So the major disadvantage of HF calculation is that the electron correlation is not taken into consideration. That's what I mentioned just now. So that's why the Hartree-Fock theory is the very basic one, or the very first level of or simple form of quantum mechanics calculation in quantum calculations. So now we have ab initio molecular orbitals method. Let's check about them, what we can do in it. So these approximations mostly we used in ab initio calculations. Now you know ab initio calculation is only theoretical calculations are our theoretical model. We are not using any experimental data in it. So what we have then, we have here central field approximation. Now this is the kind of level of accuracy we can approach in ab initio method in under quantum mechanics to resolve the problem under computational chemistry. What is that? It integrates the electron repulsion. Now, because the electron, we know that in an atom, the electron has a positive spin, the negative spin, you know, when we put in the plus arrow up or down, because they have a different spin. And if they are different spin, they may get interact or repel if they are interacting with the other orbitals. So this particular one consider deep into it, the electron repulsion. That means they are pushing them each other away from each other, and then give an average effect instead of an explicit energy. Rather than giving an accurate energy level there, they are considering that approximate results there again about the energy of the system. Then we have another approach is linear combination of atomic orbitals. This is used to describe the wave function, and these functions are then combined into a determinant. It's a mathematical term in differentiation and integration. You may consider that we are considering these functions and then we are integrating them to get some determinant to the some conclusion or single calculation to explain the system. Correct? Understand? This allows the equation to show that an electron was put in orbital, but the electron cannot be specified. So there is another drawback is that these equations put electrons in the orbital, but they cannot find out where the electron is. So that's the probability of finding electrons in an orbital. That was a drawback, but somehow since they are considering the different atomic orbitals, so calculation or approximation for a molecule is more accurate than one semi-empirical one. Then we have density functional theory, and which is very popular these days in drug discovery also. How? I will explain. First, we understand what is density functional theory. It considers ab initio method, but it's different from ab initio methods because the wave function is not used to describe a molecule. Instead, the electron density is used. I mentioned before that like CI and the MP and the density functional theory, they are considering the electrons, not the wave function of the particle or the wave function property of the electron. Rather than they are considering the electron density as single point or single charge identity or something like that. So there are different types of DFT calculations which exist now. We have local density approximation. Local density means that they are considering the local electrons, but not the outer shell electrons and all. So this method is fast, give less accurate geometry, but provide good band structures. So all the models, computational models, each model have its drawbacks. So these some drawbacks related to is that it gives less accuracy but it considerably gave approximate band structures. Then we have gradient gradient corrected one. So what happens here, it gives a more accurate geometry because it considers more approximations or more density of the electrons in the equation. Then we have the hybrid one, which are a combination of DFT and HF. In the HF, Hartree Fox, it considers only the nucleus, not the electron. So in the hybrid one, what happening then, now DFT they are considering electron, HF they consider nucleus, then they merge together and bring it as a hybrid method. So it gives a more accurate geometry of the system or the model. So DFT methods takes less computational time than the HF calculations and are considered more accurate because it is considering both electron and the nucleus property of the molecule. So in a total, in a multi-body, if we are calculating all these things, then we can exactly tell the behavior of the molecule, or we can tell the geometry of the molecule, and we can calculate the different properties also. So now we comes with the basis set. As in molecular mechanics, we have force field. In the quantum mechanics, we have basis set. In the molecular mechanics, what happens there? We have force field, and then these force fields are getting upgraded. Similarly here, these basis set get upgraded to achieve the accuracy. How? Let's see. In a chemistry, basis set is what? A group of mathematical functions that are used to describe the shape of orbitals in a molecule. So in quantum mechanics, what we are dealing with electrons, and as I'm repeating again, in the quantum mechanics, we are dealing with electrons, and the electrons exist in orbitals. Okay, so we study the orbitals. If we study the orbitals, we are studying the electrons. So if we are studying the electrons, then we can exactly figure out the chemical properties and other properties for the system or the molecule. So each basis set is different group of constants using the wave function of the Schrodinger equation. So different constants, for example, energy, Hamilton equations, and then potential energy, these different constants are included in the different basis sets, and basis on that, these basis sets are changed to higher level of accuracy. So the accuracy of the calculation is thus depend on the model. What model we are considering? Under quantum mechanics, semi-empirical, or ab initio, or our DFT hybrid method. Under DFT, we have 3: hybrid also, localized also, and then other constant one, connected one. So based on that one, we can achieve the more accuracy and then less time also. So again, but what happens in the basis set? There is a trade-off between accuracy and time. If we are choosing the higher basis set, then we are choosing more computational time. If higher accuracy, more time. If you have more time, then you can opt for higher accuracy. For example, the larger basis set will describe the orbitals more accurately, but take longer to solve. I will explain them if I have put them in the slide. For example, in the basis set, if we are using normally in the organic molecule, we use B3 lib with this 631 base set. So what is that? It's explain about the behavior of the molecule in a particular level. But if we want more accuracy, then we may jump to like B3 lib to 6311 with polarization parameter. You may have seen if someone has done this quantum calculations or DFT. So, but that one, the time will increase, the titrations level will increase, and then accuracy also will increase. So general expression for a basis set, we see that n into E is equal to negative R into R, where n is the normalization constant, alpha is the orbital exponent, and R is the radius of the orbital in angstrom. So this is the approximation we can normally use for expression of the basis set. So example here. Now we have an example to make you much understand that how we can do and what basis set is and what they consider in it. First example is a very simple one, STO3G basis set. It's a very simple basis set and use the minimum number of functions to describe each atom in the molecule. That means it's only considering, for example, hydrogen stretching, torsion, potential energy, van der Waals energy, or it's ignoring London forces and all. So it's a very simple one. For example, we use this system, and these are experimentally validated by many theoretical physics and then computationalist people's working in this computational chemistry. For example, if we apply for this nanosystem, a nanotube system, that means the hydrogen is described by the function for the 1s orbital, while the carbon is described by a 5 function. 1s, 2s, 2px, and 2py, and 2pz. You may be wondering what is 1s, 2p, but I showed you in the overview of the chemistry. This is the electronic configuration. The electronic configuration describes the electron distribution in the orbital in the different layers of a molecule. So this particular set consider the distribution of electrons like this in a particular orbital. For example, in the s orbital, in the p orbital, if we look in the p orbital, it divided the p orbital into 3 rather than considering only one as a one entity, but this basis set considered p originally as it exists in y, z, and x, y, and z axis in the space. So this basis set split the balance basis set. Split balance sets use 2 functions to describe different size of the same orbital. Sorry, this is a different type of basis set, that is split balance basis set. It used then 2 different functions. What are they? That it split the valency of the hydrogen described by 2 functions, for example, and then the C would be described by 10 functions. So 2 functions mean that in the hydrogen we have only 1 electron, so it considers the both spin of the electron. For example, positive spin and then a negative spin, it considers both the spins and then do the calculation. But for the carbon, it do more calculations, are considered 10 functions for it, similar to the upper one, like we mentioned 1s, 2s and all. Similarly, it considers 10 functions. Then we have in this one we have 631G and 6311G, which use 3 functions for each orbitals, where triple split valence set. They split the valency of the electron. Why we said that this valency? Why? Because the valency of the molecule is going to participate in the chemical reaction, and based on the valence shell of a molecule, we define the chemical property of a atom. So that's why they consider the valence set here. Then we have polarized basis set. Polarized basis set means then we are considering the charge in the valence also. So these are more accurate. These basis sets are more accurate by allowing the shape of the orbital to be changed by adding orbitals beyond that which is necessary for an atom. So how we can do that one? They are considering the charge also, and then they are considering the orbitals. Possible orbitals may exist in an atom when uh, orbitals are there. For example, now we have hydrogen only one, but they are also adding the second uh, s orbital there, and then checking how it is affecting the hydrogen property. For example, if we adding n1, then they become hydronium. So similarly, something like that, uh, they try to uh, achieve more accuracy in a system with simplification, with more simplification but more accuracy. For example, 631G, known as 631G a star, because the star means here they are adding the polarized function, and they add a D function to the carbon atom here. Correct? So this is the example or overview on the basis set. These are the basis set we used in the quantum mechanics to understand the behavior of the molecule through electron properties. So quantum mechanics, where we can use the quantum mechanics. Quantum mechanics methods are suitable for calculating, we know before, but we just summarize them here. The molecular orbital energy. If we know the molecular orbital energy, we can calculate the bonding, how these molecules going to bond with each other and which kind of bond they are going to form, and what would be the transition state of the molecule when it is going a chemical reaction. I did not mention, but we can even calculate the this reaction and transition state also if we are calculating the time independent density functional theory calculations. So that means if we have this energy from the orbitals, we can calculate these reactions also. Then we have heat formation for specific conformation. That is, we have endothermic reaction or exothermic reactions going to happen when the two molecules are coming together and forming the product, or reactants coming together and forming the product. So that calculations can be done here, and based on that one, we can optimize our experimental setup. Then we have a dipole moment. Dipole moment is the positive and negative charge polarization in a molecule. And so we can even calculate that one, and how this polarization going to change the conformation of the molecule. We can do that one. Bond dissociation energy. If we have a system and then we want to know how much energy need to be put in the system so that it broke down, we can do that one also. For example, if we are working with some for example, let me put like this, that in the natural resources, they are very big molecules. For example, tannin, or we have proanthocyanin, but this tannin and proanthocyanin make up of epicatechin and catechin. Those are more pharmacological effective compared to these big molecules. Why? Big molecules cannot inhibit the protein. The pocket is very small compared to the size of these big molecules. But if we want to break them into small one, and then we want to check how these for example, proanthocyanin broke down and then two molecules produced, but how much energy need to be put on this carbon and the oxygen bond there on the earth or the para position, because in the proanthocyanin we have different types based on the bonding between the carbon and oxygen. We can calculate these bond dissociation energy, and corresponding to that we can apply, and then we can perform our experimental we can perform our experiment, and then we can have our desired molecule for as a drug. Or let me tell you that this proanthocyanin are being used as a drug, which comes in the body getting digested by the enzymes, so and become drug. So We can even figure out, we can study that how these enzymes are breaking these proanthocyanin into smaller molecules. And if you want to understand the mechanism, the chemical reaction mechanism or the enzyme reaction to this conversion, we can use quantum mechanics. Like I said before, transition state geometries and energy. For example, A is reacting with the, no, I will say like apicatekin and catechin coming together and forming the proanthocyanin. How they are forming the proanthocyanin, what are the intermediates they are forming. I can do this reaction with the quantum mechanics. I mentioned that time-dependent density functional theory with additional parameters I have to reconsider. Using these parameters, I can do this calculation, or I can say that I can calculate these transition state geometry and energies of the system. So these are the broadly applications we can use quantum mechanics, and to understand the complex behavior of the system. System means here our protein or our molecule or our drug conjugated with the target protein. So we can understand that one. So if we say that if there is a drug and we want to understand how this drug is inhibiting the protein, we want to figure out what exactly is happening there at the electron level. So we can do that one using the quantum mechanics. Why? Because we can understand that which bond is going to form there and with how much energy. So we can use quantum mechanics there. So what's the difference between then quantum mechanics and molecular mechanics? Quantum mechanics correctly describe the bond breaking and bond formation, and even calculate electron density. But in the molecular mechanics, we does not properly describe the bond breaking and bond formation, and hence we cannot calculate the electron density. Electron density, that is molecular orbitals. Then the application of quantum mechanics is limited to only hundreds of atoms. Why? Because we know very well that if we want to have a very accurate results, 100% accurate results, that we can apply only to hydrogen. Why? Because it has only one electron. But if we are applying to the higher or multi-body systems, then the this number of atoms is increasing, and then we cannot use it. So that's a drawback. Then the molecular mechanics, uh, uh, we do not have such a drawback. Why? Because in the molecular mechanics, we can treat the atoms as macromolecules. We are not talking about the electrons. So we can consider more than 10,000 atoms in under molecular mechanics. But if we look at the quantum mechanics, quantum mechanics is computationally more expensive, but computationally we see the molecular mechanics is comparatively less expensive. Then we have in the quantum mechanics, it's very expensive. We need very expensive high configuration systems, a computer, I mean, and then we have to spend lots of money. But compared to quantum mechanics, molecular mechanics, we do not need too much high configuration computers and time. Then we have quantum mechanics, which is highly suitable for the molecule, but molecular mechanics is also somehow suitable for the small molecules, but not with high accuracy. So if we want to study the quantum mechanics, what are the programs we can use? Then these are the list of the molecules. We have this Gaussian, we have chemist, we have Molcus, Molpro, QM, Mopac, this WASP and Multivin. And these are the some of the molecules for some of the software. For example, Gamma is a free one, academic version. You can use it easily, free package. Similarly, we have this Quantum Expresso, but its only small portion is free and rest is become now paid version. But Abinit, you can use Abinit for these quantum calculations, and you can easily use them for your drug discovery projects or some similar programs. If you want more information about the basis set, there's a link. You can check it there. Then now let's come to the drug discovery phases. How we can apply now this quantum mechanics and quantum mechanics, and then molecular mechanics in drug discovery approach, and where we can apply it. So typically what happens in research on this project, drug discovery project, initial phase is this target discovery. Then we have assay development. that comes under clinical one. Then we have lead identification, and then we have optimization. Although we can use the computational methods, for example, virtual screening and all, for the identification of the compounds, but some companies do not prefer because they have automated screening process in vitro culturing and in vitro assay. They apply that one and then do the screening in a parallel approach to computational one also. So if we see in the target discovery, what happens in the target discovery, they identify the target from the literature or in a house research that they have already identified it. They validate the target with the knockout animals and sRNA experiments are tool some compounds. So they first validate the target or the drug target. Then we have this assay development. They define the assay, what kind of assay need to be done for it. For example, we are going to use enzyme, uh, or we are going to use cell lines expressing the protein, or biochemical assay, or optimization and automation for high throughput screening. Because in high throughput screening, we are going to use millions of compounds to be screened in a one shot rather than doing it in 96 well plates. So that's high throughput screening. Similar high throughput screening we also have in virtual screening as well, in computational tools, but it's a different one. Then we have a lead identification. From the screening, we have some compounds, like I mentioned in the initial slides. Now, based on the identification, we have to identify which molecules are more potent. For example, high throughput screening, again, hit analysis, we check the how the compound pharmacological properties and all. Then we select the promising chemical matter to modify further, or the whole molecule. Then we check the compounds, some properties like pharmacological properties and all. And then we check also structure activity relationship with the other molecules compared to them. If we have some similarity, and then we can use the same molecule to some similar disease. So these things we study under generally lead identification. Following, we have then lead optimization, because our target is different, or maybe we can say that the protein is getting mutated all the way during the disease, like I mentioned under the TME, tumor microenvironment. So we need to modify the molecules. So we need to do some tests that, for example, we check the activity, or we call it affinity. We select this, we check the selectivity. We also do the pharmacokinetics, that how in it is behaving with respect to time, what effect is doing on the protein, and is it stable or not. We do in kinetics also, we do metabolism also. Then we have chemical manufacturing control. These all things are done under lead optimization in a drug discovery process. So question comes that where we can use this computational chemistry. We can use this computational chemistry under lead identification and lead optimization. Just a second. All right. So lead optimization, where we can use the computational methods. I have taken only three examples here to make you understand better, because it's a huge process and it cannot be done in a single lecture. Examples are, for example, structure refinement for structure virtual screening and molecular dynamics. Then I have molecular docking, sorry, and then molecular dynamics and quantum mechanics. And under the quantum mechanics, we will see the ligand reactivity, how we can check it. For example, now we are looking for the target identification, and then what happens, all of a sudden we do not have any crystal structure. Similar thing happens with the coronavirus. When it was initially reported, there was no protein structure for it, but they just estimated they have genome sequence, getting genome sequence, and from the genome sequence they propose some model, protein model or homology model. So what happens during the homology models? We may propose the model using this computational tool, but how to figure out their stability or the optimum geometry? So here we can use the molecular mechanics to do it. For example, we just I have put example here for you. that I done it myself. For example, I use NSP14 and 7 MTAs. Till now, there is no crystal structure, I believe so, for the SARS-CoV-2. You can see that it is before the molecular dynamics. That is the homology model I used here. And then the second one after the molecular dynamics. You can see that what happens to it. So if you see here that on the N terminal, there is not much changes happening, but only the loops bit changed and the domain only shift a little bit. But and the C terminal, there is lots of changes in happening in the beta sheets and then the loops also, and then this helix loops also. So what is happening? That's mean that before the molecular dynamics analysis, the protein do not have a proper conformation. That means after the molecular dynamics, it have already achieved its original or optimum position or optimum conformation. Why it need to be done? For example, now since the conformation changes are happening on the C terminal and the N terminal, but not on the active pocket lies somewhere in this orange helix loops and yellow. So why we need to do it? For example, now we have a homology model, and then we are not defining our structure. So we are directly taking it for the docking analysis, and what will happen then? That we have a good results on binding results. We have very good results, or we have a very good docking energy. But when we put it in vitro one, then we do not have any positive results there. So maybe why? Because the active site conformation was changed there. So because the active conformation was not actually real as it's supposed to be in the molecular in the homology model. So what we do actually, we first optimize the structure and then consider for the docking, and then we can exactly figure out how many molecules going to be a positive inhibitor or going to be showing positive results. Then we can increase the accuracy. Correct. So similarly, we can use the molecular mechanics, these molecular dynamics. We can understand the behavior of the ligand at the active pocket, and then we see how much stability is there with the protein active pocket with respect to time. If the ligand is leaving the pocket during the simulation, then we say that there is not much stability, although we have a very good docking energy. But mostly what happens in the most of the cases, if you have a energy, docking energy more than -7, we consider it as a moderate inhibitor, or it should inhibit. But sometimes it depend upon the algorithm in the docking. For example, which algorithm, because different docking programs have their different algorithm for the docking. So it again change our level of accuracy. Similarly, in the basis set and all, in the docking also, we had different types of algorithm which they consider. For example, they are considering hydrophobic interactions. Some are considering, let me put an example. For example, here in glide from the Schrodinger, it is considering even the hydrophobic and hydrophilic interaction during the docking, but other programs are not considering hydrophobic and hydrophilic. So something some of the differences like this. So to understand exactly the behavior of the protein along with the ligand, we go for the molecular dynamics. And beside that also, we can, if we want to check only that during the reaction, enzyme reaction, does the conformation active site protein active pocket of the protein does change or not during the enzyme reaction, we can do again molecular dynamics. Then we have a refinement of the ligand or molecular mechanics for the ligand. For you, I have put an example here. I have done this refinement of a ligand, the same buffering I showed in the first slide. You can see here that before the optimization, it has energy of 25.035, but after optimization, but it's not very accurate one, because I used only UFF, universal force field. You can see here that this hydrogen, the polar hydrogen on the side chain, the benzene ring is changed, shifted towards the left side. But in the original one, that is a black one, is exactly perpendicular to the plane. So you see that its conformation is changed here, and so the energy. If we are using this optimized one and then we are trying to do dock in the active pocket, then we will have more accuracy on the docking data. Correct? So this is how we can use the molecular mechanics on the drug-like molecules uh, uh, to understand their behavior during the docking, or to get a more accuracy in the docking data. So similar approach we can also use using quantum mechanics. For example, I have used here Hartree-Fock method for your consideration here. You can see the electronic energy here is minus 1022, but here is minus 28. You can see that there is not much difference, but still there is a difference. And then if you see the geometry, then there is again geometry change. For example, in the black one, that's the original one, so there is not much change in the side chain. This benzene is a shifted one towards the right side, but if you see in this one on the green side, which is optimized one, optimized geometry, you see that the benzene ring is shifted to the plane, perpendicular to the plane, and then the hydrogen this to attach to it with the carboxyl group is again shifted towards the what you call a left side. So you can see the difference here, that almost before this optimization there is not much change, but after optimization during docking and molecular mechanics there is a change. So what does it mean? That means if we are taking only the original ligand for the docking, the conformation, the interacting atoms in the ligand is not going to interact exactly with the protein, or maybe they are giving some interaction, but which is not relevant exactly in vitro experiments, and we will not have some accuracy in our docking data. So it is very important that if we are doing this docking, we optimize our ligand. For example, we can use molecular mechanics, which is normally gets done when we are using the docking. During the ligand preparation and all, this process get done its own. But if we want more accuracy, we can use quantum mechanics. Correct? Now, but there's question comes again, that can we use quantum mechanics in docking also? Yes, there is another method in the docking where we use quantum based molecular docking, or QLID. So there we are using quantum mechanics for the docking of the molecule to the protein. But this process is very expensive and computationally expensive one. So if we have some compound collection of molecules like 50 to 20, and then we want to check it using the quantum QLID docking, we can do it, or else there is a problem. Why? Because it will take more time, or maybe more than 1 year, something like that, and then we do not have that much time. Correct? So yeah, this is the docking. Now then we have the molecular dynamics. So where we can use the molecular dynamics in lead optimization, we can use it to investigate the flexibility of the protein. I mentioned in the previous slide, we can check the conformations of the active pocket. Does it change or not during the catalysis, or during the reaction, or when it's binding with the ligand? So there is a change or not. So we can check it. This flexibility of a protein. Then we can also search for the putative binding site, which are not obvious or not present in experimental data or in the experimental structure. Putative binding site. There is another approach. For example, in the docking, you are using blind docking, but in the molecular dynamic or assemble-based molecular dynamics, what we are doing, because we know that the protein is getting changed during the molecular dynamics, it's changing its conformation. So we put the ligand there, and then we try to check where it else can bind apart from the active pocket. So there's advanced version of the molecular docking version, or the screening version, which mostly industries and some high research groups used. So that's an approach. Then they use this molecular dynamics to calculate the binding energy. Binding energy during the molecular dynamics, we have different approaches. For example, we call it free binding energy analysis, or landscape free energy analysis. What happens that at every interval they calculate the energy, how the energy of the ligand with the receptor, or sometimes we call it another approach, that is FEP. This perpetual energy, some free energy fluctuation analysis. So these are different type of analysis we can do. Beside that, we have conformation sampling and analysis. We can check conformation analysis. That means how the protein and the ligand complex exist. For example, if we are doing 100 nanosecond, we can check the conformation, for example, at 1 nanosecond or 10 nanosecond or 100, something like that. And then we see, does the ligand still exist in the active pocket or not, or there's any change in it during the simulation. Change that means sense means that does any flexibility in the ligand or the protein occurs or not. We can check it also. Beside that, we can also find some binding poses, and then we check them how they are interacting. That means how, what kind of interaction is happening there. We can also calculate them. Beside that, we can also calculate the kinetics. For example, binding and unbinding kinetics can be done from the molecular dynamics analysis. Beside that, sometimes, for example, in case of some proteins, the water is required at the active pocket of the protein, which initiate in the catalysis. For example, in human MMP1, matrix microprotein 1, which is a collagenase degrader, it need a zinc ion at the active pocket along with the water for the catalysis of collagen. So if we want to analyze this, we want to check the water structure there, how the water is going to interact with the substrate, and how is contributing in the reaction of the catalysis. We can also understand it from the molecular mechanics. Yes, that's all. And then we have molecular mechanics. There are many calculations we can do the molecular mechanics, but for example, we can do geometry optimization that I have already explained. But another thing is that we can calculate the LOMO and HOMO for the ligand, and then from this LOMO and HOMO, we can calculate the reactivity of the ligand. How? For example, you can see here, I have two flavonol, 1 and 2. I have calculated the LOMO and HOMO, and then corresponding to them, I have calculated their difference, LOMO and HOMO difference, that is a delta G. I have converted the value into electron volts. So if you see here that I have the flavonol 1, it has a energy gap of 5.2, and but in case of a gap is 1.6. So what it indicates? Energy gaps indicates that how the let me tell you first here that what is LOMO and HOMO. So HOMO is high orbital, high occupied molecular orbital, low unoccupied molecular orbitals. So that means the electrons will move from the HOMO to LOMO. There's an interchange between. So if their energy gap is less, then the electron transportation can easily be done, and then the molecule become more highly reactive. But if the transition of electron is less, then molecule is less reactive. So in case of flavonol 1, you can see that the energy gap is more than 5, but in case of flavonol, the energy gap is less. That means they can easily jump from one orbital to another orbital. That means LOMO to HOMO, they can easily, or HOMO to LOMO, they can easily move, but in case of one, is not possible. So based on this energy gap, we can calculate the behavior, or we can say the reactivity of the ligand. So under quantum mechanics, during the process for lead optimization. Why we don't want ligand which is reactive? Why? Because if it's reactive, highly reactive, then it may cause toxicity in the body. It's rather than acting with our target protein, or our target drug target is reacting with the other targets also. So this is why we calculate these calculation on the quantum mechanics for our ligand. Well, this is all for our today's section. And if you have any query, you can write me, or you can directly ask me. I'm available to answer you. Yeah, thank you.
Thank you, sir.
Thank you, ma'am.
If anyone have query, so please concern with the Dr. Bharadwaj, sir. Sir, there one question by Altaf Shah.
Yes.
Sir, please explain how the pulling force is generated there. Mr. Altaf Shah, you can unmute yourself. and directly discuss with Dr. Bharadwaj sir.
Hello,
Hi.
hello, sir.
Hello, Altaf.
Good afternoon.
How are you?
I'm fine, sir. Sir, actually, I want to know when we are going to perform the visual molecular dynamics, and there we are during the formation of complex. There are some forces which arise there. But one of the forces is pulling force, and what actually it means?
Pulling force.
Yes, sir.
Actually, what happens that you need to understand what happens during the molecular dynamics.
Yes, sir.
Molecular dynamics is what is happening there. We are providing the external force, correct?
Yes, sir.
So when we are providing the force to the molecules are going to vibrate, correct?
Yes, sir. Yes, sir.
Why they are going to vibrate? Because they are loosely bound to each other, and under
Yes,
the
sir.
force, of course, they're going to move around. But sometimes what
Yes, sir.
happens, because of the net charge, they either attract each other or they repel each other.
Yes, sir. Sure.
So what happens then, when the molecules are under vibration or under molecular dynamics are under force, it shows its behavior. For example, now you are pulling something, and then because of its now have a net positive or negative charge, or they have a
Church.
like charges, so they
Yes,
either
sir.
repel or attract each other. So in
So they
case of
will
you are
repel.
talking about pulling, so I understand that you are talking about that they are repelling each other and then they coming and vibrating each other. I mean,
Yes, sir.
vibrating on the particular space. You
Yes,
mean
sir.
that pulling energy, right?
Yes, sir.
So it comes.
What's the energy behind it? Actually, I want to know that what is the energy behind it that pulls or that stretches the bond that is between that actually present between the atoms.
So that's what I'm explaining you, that under
Yes,
molecular
sir.
dynamics, what we are doing, we are providing the external energy, and we know that they have a certain parameter. For example, bonding, stretching, and this what you call torsion and all. So what happening
Yes,
there?
sir.
Then when you provide the energy, they either get interact or repel. So
Repel.
exactly
Yes.
what energy is acting in your system, if you want to
System.
understand that one, for example, now there are various energies in the system like van der Waals or like hydrogen, hydrophobic and all. But if you want
Others.
to understand which energy is doing that one, then you have to go for ab initio molecular dynamics. So what
Dynamics.
happens in ab initio molecular dynamics? You are considering the different forces, including the charges. When you are including the charges, then you can exactly figure out the behavior of the system, correct?
Yes, sir. Yes, sir.
So under these
Yes, sir.
conditions, then exactly you can figure out which force is responsible for pulling in your system.
Yes, sir.
Understand?
Yes, sir.
As
Yes,
such,
sir.
if you
Got it,
want to
sir.
say that, as such, if you want to tell that which energy uh, uh, is responsible for pulling in your system under simulation, you cannot, because you do not know anything about your system, right?
Yes, yes, sir. Yes, sir.
Until
Got it,
unless
sir.
you don't until unless you know what kind of energies are there, and what is more impactful there, I mean to say, which is at more how to say strong, or which is strong and which is weak there, and based on that one you can calculate the net pulling energy. But at least just random one you cannot understand.
Yes, sir. Yes, sir.
So
Got it,
you
sir.
need to understand the system there.
Yes, sir. Thank you,
Yeah,
sir. Thank you very much, sir.
you're welcome. Any other question?
Ruchita.
So Dr. Vivek Dadvedi,
Yes,
Dr.
ma'am.
Vivek Dadvedi, sir,
Yes.
Hello.
sir,
Ah,
Now
sir,
it's
आपने
clear.
भी
It's clear to me.
okay,
Dr.
okay, okay,
Bharadwaj had dictated all the things.
okay,
Okay, okay.
okay, sir.
Okay. Thank you. Any other questions? Anyone else from the panel or someone else and participants would like to ask me? Uh,
Okay,
anyone?
sir.
I believe
Sir,
so.
actually,
Everyone understand little bit. I tried my best not to include any mathematical equations and try to give an overview, because if we are going to jump inside and if we want to look inside what is happening, it's very complicated to explain. I believe so that everyone understand the basic concepts of computational chemistry, including this molecular mechanics and quantum mechanics, and hopefully we do some future lectures similar like that, and then we can exchange our knowledge again.
sir, thank you,
Yeah.
sir, for this
Okay. Thank you so
informative
much.
lecture.
Thank
Thank you,
you.
sir.
Thank you for the given opportunity.
Thank you very much, sir.
Thank you, Altaf.
Ruchita,
जी sir. जी sir.
can you summarize the program?
जी sir. So this is the very wonderful lecture on molecular dynamics and its importance for drug discovery. This is very useful to us for enhance the knowledge in this drug discovery field. Bharadwaj sir, just सर नहीं आपको नहीं बोल रही हूँ सर मैं जस्ट आपका थोड़ा सा डिस्क्राइब कर रही हूँ आपने जो भी बताया है
Okay, sure.
जी सर. Actually, Dr. Bharadwaj described the molecular dynamics is a computer simulation method for analyzing the physical movement of the atom and molecule. The atoms and the molecules are allowed to interact for a fixed period of a time, giving a view of dynamic evolution of the system and quantum mechanics also. So this is a very wonderful lecture. At the end of today's program, thanks to all of you for your participations and support for making this workshop successful. Tomorrow in the last day of the workshop, Dr. V. Muksing will deliver his lecture on the computer-aided drug design method and case study. So tomorrow's session will start with the sharp 12 o'clock. This is my humble request to you all. Kindly join this online workshop 5 minutes before. Now I need to permission by all the invited delegates and organizing committee members for close today's session. Sir Shukla sir.
Yes. Yeah. I would like to thanks to our resource person, Dr. Bharadwaj, and organizing team, teacher and staff member and all participants. I am a teacher of social science, but very patiently listen to this program and also get new information. Tomorrow will be the last day of the workshop and we will meet again at 11:50 a.m. Thanks, Ruchita.
Thank you, sir.
Can I end the program?
Yes,
thanks,
sir.
Bharadwaj again.
Thank you, sir. Thank you so much for the given opportunity. Thank you.